Thank you, John. Well, there's a lot of that John just covered that started off mine, so we're going to kick right into it. So as John said, I came to Morgan Cure Paralysis. Actually, David, I don't think I made the cut on the pictures, but I played in the golf tournament for many, many years prior to joining the board. And as John pointed out, when you go through that and it's in your family, it is it's one of those moments where you have your life before that and then your life after that. And John's right. Went out there, that was the first place we started looking. My wife and I sat down and was going through everything. We got more information. And information gives you comfort too. And it's something we really needed at that moment. So John's right. The tournament was about a month after the accident, and I came up to John and asked him, what can I do? What do we do? And he helped me through that. So thank you, John, and thank you, Peter, for giving me an avenue to help. With that, Dr. Blackmore, how did you come into spinal cord research? What drove you to that field?
Yeah. Well, all right, before I get to that, let me just say a couple of things. First of all, it's amazing to see everyone here. It's a Tuesday evening, and you're here to listen to a guy talk about science. That's strange, but amazing. So thank you for that. I guess I also want to say to some of you, I'm kind of sorry you're here, because you're here because the injury touched you or someone you care about. So you're here because you would like to do something about that, so kind of sympathy and respect for that. And I say all that to say this, I'm in the same boat. So I came to this because my mom had a spinal cord injury. There was an accident back in 1987, sort of did a lot of damage to the family. I walked away, but she came out with a high cervical injury. So I kind of grew up seeing what that looks like up close. Didn't come straight to doing research. I sort of wandered around, but in my mid-20s I realized that, I realized a few things. One, I wanted to make a difference, like a lot of people do. Two, I was never going to be like a politician or a policymaker, but I was kind of good at figuring things out, so I could make a difference there. And three, there was this problem, right in front of me all this time. So ever since then, I've sort of been aimed straight at the problem of spinal cord injury.
Thank you for that.
Yeah.
So let's kick it off with your research. We've got some questions, but could you just start us off to give a general overview of what you're doing and what your focus is?
Okay, yeah. If you don't mind, I'm going to kind of frame the problem, if you will, sort of describe what we're trying to do. So, and to do that, I need to lay some science words on you, but there's just a couple, I promise. In a nutshell, what we're trying to do is we're trying to repair the long connections between the brain and the spinal cord. So I think everyone here probably knows there's cells in the brain, they're called neurons, and they have what's called the cell body. It's kind of like the center mass of the cell, and it lives up in the brain, and that's like where the DNA is. It's kind of like where the cell is, but it has this long projection that goes down the spinal cord to send signals. And that thing's called the axon. I'm going to be just using that word right and left this whole evening. So axon, that's the thing that gets cut in the injury. That's why the signal stops at the site of the injury, and that's the thing that we're trying to regrow. What you have to understand about that is just the scale of things. So let me illustrate it this way. Let's pretend that my body is the cell body. So I guess we're in the brain right now, right? And then my arm is the axon. This arm is really long. The axon is incredibly long. It would stretch about from here, I did the math, to about downtown St. Paul, okay? So there's long things. So most of my mass is out there in that cable, or the axon. So after a spinal cord injury, the axon gets cut, and what we're trying to figure out is a way to convince the cell to regrow the end of the axon, extend it all the way back to downtown St. Paul, and then make the correct connections once it's there.
Excellent. So why is it so hard for the central nervous system to heal after an injury compared to other parts of the body that do heal when you have a cut or something like that?
Yeah, okay. All right, so two two reasons it's so hard, and then a reason for optimism. All right. Reason number one, I already alluded to, we're asking the cell to do something that's just inherently really, really hard. It's a huge amount of cellular mass and a huge organizational challenge to convince this cell to regrow such a long, long process. There's no other cells in nature that have any kind of anatomical challenge that big. So that's one problem. It gets worse. At the site of injury, there's a whole host of separate problems. So just to really stretch the analogy, if you think about my arm growing all the way to downtown St. Paul, maybe it's going along I-94 there, and there's been a break in the road. There's a physical cavity that forms, so you're going to have to build some kind of a bridge over it. There's a whole bunch of molecular cues. You can think of them as sort of like stop signs that are telling the axon not to advance. And then this scar tissue forms, which is a good thing, right? You have to kind of scar off injuries. But one of the properties of that scar is it stops the axon physically from moving forward. So you're asking a lot of the cell. You've got these barriers there at the site of injury. It seems like a really heavy lift. Okay, but let's take a big step back. Let's look all the way across nature and realize there's a lot of neurons out there with a lot of axons in a lot of species at a lot of ages. And you can start to look at all the different neurons, and what you realize is that many, many, many of those have the ability that we're talking about, this ability to regrow an axon. For example, there's neurons that live in your spinal cord, and they send their long axon all the way out like to the tips of your fingers to move. And if you cut those axons, they'll regrow. So even in our bodies, there's axons that can do this, right? When we were embryos, those axons had to grow originally, right? So there was a time in your life when all of your axons could grow. And then it gets even better, like fish have this weird ability, not a weird ability, they have a natural built-in ability to grow axons, and so do frogs, and so do worms, and so do flies, and so on. So this is a reverse engineering problem. For all the challenges I laid out, there are examples of how it can be done, and so we just need to follow those.
Does science have an understanding of like why the brain wouldn't trigger it to grow more?
Yeah, here's where, I mean, in a way it almost gets philosophical, but you can start to think about evolution, right? So one thing I just sort of baked into what I just told you is that when you think about axons not being able to grow, that's something that happened in evolution. It's weird, right? So evolutionarily old species can grow their axons, and somewhere along the line our brain and our spinal cord stopped being able to do that. So that's weird, right? So why? I can only speculate, but here's what I think explains it. It's a two-hit problem. Number one is once you're big enough, there's no real advantage to being able to regrow your axons, because even if you can, there's a limit to how fast you can do it. So you're sitting there paralyzed for a long time, weeks to months, meaning you've been eaten long before you get repaired. So there's really no evolutionary advantage to being able to regenerate. That's one. And two, once your nervous system is complex enough, it's probably the case that once it's wired up, you don't want to mess with it too much. There's probably a good reason to hold it in place, all else equal. And so there's an evolutionary pressure to restrict growth. And you put those two things together, and you get right where we are today.
Okay. With that, you talked about, studies how gene regulation affects axon growth. And you talked about the other species that can do it, and even the certain neurons or certain axons can grow. Can you talk about how you get to that and why that's so important in your research?
Yeah. All right, we're doing this, everyone. We're going to talk about gene regulation. But it's really not that bad. I promise it's not that bad. I'm going to come at it sideways, but I promise I'll get there. So we all know our bodies are made up of cells, and you have so many different kinds of cells in your body, and they're doing crazy different things: heart cells and pancreas cells and neuron cells and skin cells and on and on and on. And they have different shapes, and they do wildly different things. But here's the thing. They all have the exact same DNA, and they all have the exact same genes in them, right? A gene is just a stretch of DNA that the cell uses to make something, and you've got tens of thousands to hundreds of thousands of genes. But the point is, all the cells have the same gene, so how can that be, right? How on earth do you take the same DNA and do such different things? The answer is each individual cell is choosing to activate or inactivate a different set of genes. It's sort of selecting from those 10,000s and pulling out the ones that it's going to use. So what I'm saying is, at a really high level, if you want to understand why cells do wildly different things, you need to understand why the cells are activating some genes and inactivating other genes, right? So now you see where I'm going with this. What I think is that the behavior of growing this long axon, this long cable, is so radically different from just the neuron living its life and sending signals that you're in the realm of having to think about the activation and inactivation of probably hundreds to low thousands of individual genes to achieve such a fundamental shift in behavior, right? You're not going to get there by tweaking 1 gene or 2 genes or 10 genes. It's going to be hundreds, right? And so that's why, well, so one, let's just recognize that's kind of a radical proposition. What I'm saying is you're probably not going to get there with like 1 compound. You're not going to kind of tweak your way to repair. You're going to have to sort of reprogram the cell at a massive genetic level. And I guess full stop. So that's why the lab is so focused on gene regulation, trying to find tools that can shift hundreds to maybe thousands of genes up or down in a way that reactivates the ability to grow this long axon.
How do you go about finding that right combination? How is that accomplished?
Yeah, here's the cool thing. So here's the benefit and why it's the most exciting time to be me right now today. In the last, I'm going to say 10 years, maybe 15 years, but 10 years and then 5 years for sure, there's just been an explosion in the ability to look at gene activity in cells and get a really precise readout across tens of thousands of genes, which ones are on, which ones are off, and how much they're on and how much they're off. And what that means is that kind of as a species, as a scientific collective, we've been working our way through all these examples of cells neurons that can grow their axons and neurons that can't grow their axons. So now you start to stack up data set after data set after data set, and what you can do is you can line up all the regenerators, the successful growers, and all the non-regenerators, and you can start to find the patterns. You can start to say, okay, a lot of this is noise, a lot of this is just irrelevant differences between cells, but some of the constants are there. And so when you do that, it turns out there's several hundred genes that if a neuron is going to grow its axon, those genes are going to be massively up. And then there's another smaller set of genes that are going to be massively inactivated because they're probably getting in the way of growth. And so for the first time, like we have a signature, we have a target, like these are the genes that if we can just turn up or turn down, we think we can start to reactivate this growth.
Oh, interesting, fascinating. But you talked about, so you can start regenerating the growth, but you've also said that the axon stops at the scar. So how do you bridge that, the gap between the two?
Yeah, totally. And it's funny because that question is where the field started and where the field was like 20 to 30 years ago. At the time, it wasn't really appreciated that there was this kind of genetic problem inside the neurons. The attention was really focused on the injury site itself, naturally enough, because you have to solve that problem. But the good news is there's been tremendous progress on finding like biomaterials or different cell types, things that you can put into the injury to basically build a bridge to give those axons something to grow on. So I'm going to say like huge progress, maybe we're even there, like maybe we're there collectively of being able to put something in the injury site. We've also made other people in the field have made huge progress. And remember I said there's like these molecular stop signs that are present? Well, they've been identified and sort of knocked down. So a lot of the barriers to growth have been taken care of, although there's sort of one outstanding barrier to growth that we can maybe talk about, or not.
Let's talk about it.
Well, it's the scar. It's the actual scar tissue that forms at either end of the injury. So I'm not going to drag you into like the real scientific weeds. People have appreciated that a scar is there. I think we kind of had a false lead on the source of the scar and what to do about it. The field went down that road and realized, oh, that's not maybe the real problem. There's this other cell type, these fibrotic cells. Oh God, I'm in the weeds. Help me get out. There's a thing called the fibrotic scar that I don't think anyone has a really good handle on, and it's probably one of the places that I'm hoping the field can kind of push on in the near term, because it's a real problem right now.
John had talked about AI and about the things it can accomplish in other fields and the like. Do you think there's any applications to this? Does it help accelerate your research? Does it amplify it, bring things forward?
Oh yeah. Okay, I'll answer in two ways. First of all, just sort of what is AI doing in the lab? It's a daily tool now, and it's really in two main ways. One is to analyze all these data sets, you need a lot of software and coding and so on. It turns out AI is really good at coding, like crazy good at coding, to the point where like a true novice coder like me can just ask AI, like, here's the task, and it'll spit out code. And it's been this huge accelerant to our ability to handle the data. And then I think we use it just like everyone else does. I come across a new gene, I don't really know what the gene does. I just ask AI. It'll summarize for me, really nice, clear, way clearer than I can be, to be honest. Why am I here? We should just ask ChatGPT and it'll just tell you. But so that's kind of what it's doing in the lab. I want to give you a little mini story of what it might be doing for the field. I happen to be consulting for a company right now that's trying to use AI to address spinal cord injury. I can't say too much, like non-disclosure agreement and all that, but at a high level, what they're doing is really clever. They're taking all this genetic data that I was talking about that sort of builds a signature of growth versus not growth, and then they're having AI look through compounds, like drugs, that have already been approved by the FDA that might have the property of sort of mimicking this signature, right? So maybe, just maybe, the AI can spot a drug that's been approved for some other indication, but which might be beneficial and help neurons grow. And so they've gone through this process. They've developed a candidate list, a short list of about 70 compounds. I'm currently going through them manually to kind of see what makes sense and what doesn't. We're going to meet later this week, and maybe AI will have succeeded in finding something that already exists that could help people really, really quickly.
Oh, that's encouraging.
Yeah.
Very encouraging. So you have your field, and there's many other tracks on SCI research. And can you talk about some of the other research out there that really excites you?
Sure. Yeah. I'll two things, I think. One I already mentioned. It's just the tremendous advances in biomaterials to solve this problem at filling the injury gap itself with something useful. So huge progress there, and I'm very excited about that. The other, the big source of excitement in the field is totally separate from my research, but I think worth mentioning. The reality is most injuries, anatomically, physically, they're incomplete. It turns out that even clinically complete, meaning complete paralysis below the injury, there's almost always at least a few axons that are still there, kind of sneaking through, right, that weren't taken out by the injury. And the reason that's so important is that it turns out you can use that little bit of remaining axons as a substrate to do some really helpful things. And what I'm trying to get to is this idea of neuromodulation or electrical stimulation. So many of you have probably heard of this. If you apply electricity to the spinal cord below the injury, it can take those axons that aren't enough by themselves to have actual function, but you can kind of amplify them. You can turn up the signal, and now people are starting to get function back. And like, it's small, but it's so important. Things like better bladder and bowel, or even in some cases, I've talked to people who have gotten pincer grasp back after spinal cord injury, and that is huge. If you're living without the ability to pincer grasp and then you've got it as a result of the stimulation, that's a life changer, literally. So a lot of excitement there.
Yeah, remarkable. For your research, if you could have one breakthrough, what would you choose?
Only one?
We'll start there.
All right, fine. I'll do. Okay. It's hard not to. You know, I'm going to take that as like the thing that I need for my research. But here's what I'd say. What we're trying to do right now is we're trying to look at a neuron that can grow an axon, look at its genes, and then a neuron that can't, and sort of shift its gene pattern to resemble the one that can grow. That's the goal. But the way we're trying to do it, it's all about these weird constraints. We can only deliver like 1 or 2 or maybe 3 genes at a time. So we're constantly looking for these like master regulator genes that you can send in one gene and it'll have this ripple effect that will kind of get you to where you want to be. But my breakthrough technology would be, okay, I'm going to get science fiction for a minute, like, cause just to illustrate, this is a fun question.
Yeah.
I have no idea how you would do this, but wouldn't it be cool if instead of like trying to send in a specific gene, there is a way to somehow just like read the code, like the overall code of that cell that could grow, and somehow import it, right? So if you could, almost like a computer program, instead of going gene by gene by gene, if you could somehow get the full constellation and then move that information into the new cell. I have no idea how you do it. What I'd settle for is give me a technology where I could push 100 genes around at a time practically, instead of trying to find that one that's going to be the domino to knock all the other dominoes over.
Oh.
Yeah.
Cool.
That's what I want.
Sounds good to me.
Anyone have that?
Let's switch gears a little bit here. The government is looking to cut back on research funding across the spectrum. How is this affecting your research and just research in general at universities?
Yeah, I mean, what can you even say to that? Just to give you a feel for the scale here, until recently, there was a program through the Department of Defense that was earmarked for spinal cord injury. It's a little shy of 40 million a year, and that is gone. It doesn't exist anymore. So that just that is a pretty massive hole. The NIH cuts are happening on 2 levels. One is really public. There's kind of this public fight between the administration and Congress. Congress is actually pretty supportive about the overall budget, but meanwhile there's this kind of quiet squeeze where the NIH is changing its rules on how money can be like carried over year to year, and so basically they keep sweeping funds, and that's been really affecting my lab and many others. Honestly, the immediate effect is, I'll come back to this later. Here's what you want in your scientists. You want your scientists taking risks, trying new things, and pushing the boundaries. When everyone feels squeezed, they stop doing that. They stop hiring, they stop innovating, and that's already happening. Everyone's holding in place, trying to figure out what's it going to look like in a year? Can I afford to buy that new equipment, et cetera, et cetera. So it's been a real freeze on innovation and progress already happening, even before the real money squeeze hits.
What does that mean for organizations like Morgan Cure, which literally is looking for those risk takers? And how does that impact organizations like ours? Or what can we do to help?
Yeah, I mean, pressure's on, right? Pressure's on all of us, but anything you can do to keep innovation funds flowing, to combat that freeze, to combat that hesitancy, to keep the growth, to keep the momentum, that's it. And we're begging you, literally, anything you can do to keep that going.
Yeah. And essentially, that's a call to action for Morgan Cure, for our supporters and the like. Now more than ever, we're needed, and for the obvious reasons we just discussed here. Listening to the podcast, listening to you today, it's exciting. It makes me happy. It makes me think that this can happen. I mean, for someone who wants to support spinal cord research, what would you say to them?
Well, now you've gotten to know me well enough. I'm going to say a lot. But I think this is almost it, so bear with one more, I guess, mini speech. What I wish everyone understood is sort of how research really works, and then how important this organization and organizations like it are. And here's what I mean. So research is expensive. It's unbelievably expensive, and I think everyone sort of has some sense of that. You need trained people, you need the specialized equipment. My lab runs on about $500,000 a year, and that's just one lab, and you need a lot of labs working. What that means is if you want to create research and you want to sustain research, you need either a federal government or you need like private input at sort of the Gates, Bezos, Musk level, right? That's to sustain research. But here's the thing, and this is my lived experience for the past 14 years in the lab. The big support, the big federal grants, they kind of grind forward slowly on these expected paths. And you need that. You need the infrastructure. I'm not knocking it. I'm not biting the hand that feeds. But the reality is it's slow, and it sometimes sort of ponderously moves in a direction that's not really helpful. So what you need to actually move research, to actually spark the change, is little pools of money where you can take extreme risks, try something that probably won't work by the numbers, right? But it might. And if it does, you can shift the research, you can move the research, you can jump it forward. So what I'm trying to say is, if you want to sustain research, use your vote, use your email to your representative. But if you have $1 and you want to move research, you want to actually make an impact in a lab, find an organization that's small, that's nimble, and this is the key, that is willing to take risks. Because that is how you jump the research forward. And that's not just abstract, that's been my lived experience these last 14 years. Every cool new direction, including the thing I'm most excited about right now in the lab. I didn't even get to talk about it. I think I've got the genes going where I want them to go. Where that idea came from was like small support from a local foundation, the Brian Reich Foundation over in Milwaukee, and the Morton Foundation. That's how we got to where we are. So, end of speech, but just understand how valuable, how much we in the lab need the kind of high-risk support we're getting. And to illustrate that, this is, let's just get really cheesy. This is my hat. And you probably can't read it, but this is my Peter Morton hat. And I've had it for many, many, many years. I go running in it. I wear it around town, although not much because it's kind of old and ratty now. But I wear it all the time. And I wear it because it reminds me of where I came from. I came from Minnesota. Peter Morton was the first grant I ever got as a graduate student. But it also kind of keeps me focused and helps me remember, why I'm in this and kind of what I owe to all of you to keep moving forward. So thank you is what I'm trying to say.
I guess before we go, are there any questions from the audience you'd like to ask the doctor?
Thank you, Dr. Blackmore, for everything you're doing. I really appreciate it, and I know everyone in this room does. I guess the question that I have for you is, where are the impediments right now to, from a regulatory standpoint, other things that are saying, aside from just having more money, where are you experiencing challenges where there can be things broken down that's going to make your life easier and make progress easier?
That is such a great question. It's a target-rich environment for an answer, but I'm going to pick what I think is the most important one. It's a one-word answer. It's combinations. Here's the problem. From everything I've said, I think you've got it. if you're going to actually cure spinal cord injury, it's going to be a combinatorial treatment. You're going to need something in the injury site, you're going to need something to stimulate the axons, you're probably going to need something to knock out those stop signs, the inhibitors. The problem is the system, and I hate to use that word, but it's true, there is a system. The system is not built for combinations. The commercial system is built for monotherapies. A company has its approach, and that company takes its monotherapy, and the investors are asking, is that monotherapy going to have an effect? And the regulators are the same way. You can't do an FDA trial with multiple treatments. You have to test them one by one by one. The whole system is set up for monotherapies, and monotherapies are not going to cut it. And there's no way we can work our way through the regulatory pathway for each individual monotherapy and then combine. We have to change this. We have to fight that. We can't accept that constraint.
What you just said sort of leads to the question that I had, and that is, what other areas neurologically research, Parkinson's, for example, someone who lives with ALS, is there crossover? And maybe you just answered that there can't be because of the regulatory environment. But I'm curious about where there might be something that's done in the area of Parkinson's that could affect spinal cord injuries, or vice versa.
Yeah, no, that's a great question. There's some scientific crossover. It's I won't drag you into the weeds, but we'll say.
Like the weeds. The weeds are good.
No, okay.
Your weeds have been delightful.
I guess I'll say at a high level, my kick in the lab and coming from the money that sparked this from Morton Cure Paralysis is the idea that in order to shift cells into this growth state, there's these stress pathways that need to be tapped into to kind of inform the cell that it's been damaged and to convince it to mount a response. And then it turns out a lot of what I'm learning about these stress pathways were worked out in ALS. In a very different context. ALS is trying to shut these stress pathways down. I'm actually saying, well, we don't want to turn them on too much or too long, but we might need a little bit of them. And so that's just one example of how you can take the science from one field. The other crossover is just inspiration. Other indications have done something brilliant that spinal cord injury is now doing. It's creating venture capital funds that are dedicated to that problem. They're called evergreen funds. The idea is you get venture capital, you invest in early stage companies, you try to make enough of a profit to get back into the venture capital fund, and repeat, so that you're sort of powering commercialization in your indication. And other, ALS has done this, Duchenne's disease has done this, and finally spinal cord injury is doing it as well.
I'm just curious about the role of patients. I remember back when you did the golf outing, you had Jack Jablonski as a guest, and it was pretty powerful. And my wife and I happen to know a Paralympian who's chest down, and I'm just curious about how their involvement in research is helpful, either to getting notability of treatments or actually being willing to participate in experimental treatments.
Oh, I love that question, because now I can get on one of my other soapboxes. So, how far back to go? I think academic science is great, but it has some real limitations. And one of them is this sort of disconnect from the patient population. And what I see some scientists getting sucked into is kind of science for science's sake. So that's a long way of saying, I think what we ought to do is we ought to take people with lived experience with spinal cord injury and put them in research labs as consultants, so that scientists like me are continually asking themselves, is this experiment really moving towards something that's going to be useful to someone someday, or are we just kind of playing in the sandbox, right? And so there's a program that does that. My lab's been participating for about 4 or 5 years. So every week we have people with lived experience that come to our lab meetings and sit in on the science. It actually works. It's incredibly powerful to have that partnership between the, I don't call them patients, they're not really patients, they're people that are just living with the injury. They have so much to share. Anyway, et cetera, et cetera. So thank you for the question. My lab's doing it. There's about 8 or 9 other labs around the country doing it, and we're hoping we can expand it and just make that the norm for spinal cord injury. That if you're doing research, you've got someone sitting there helping to guide the research.
You want to talk up? Can you talk loud?
This may be way off base, but with all this information with mRNAs and vaccines, are you guys using mRNA technology at all to help with the bridge of the cells through the scar tissue and such, or is that something totally different?
I love that question. Yeah. So yes, not exactly for the bridge, but this whole time I've been talking in general terms about how, you have some genes active and some genes inactive, and you want to shift which ones are active so you can make the axons grow, that whole thing. mRNAs are a great tool to do that. You can send in mRNAs to cells and you can convince them to turn on or turn off certain gene products. So yeah, it's a great question. It's kind of the tool of choice for a lot of what we're doing.
Going once? Twice? No, thank you for your questions. I think it showed everybody was fully engaged at all times. You're talking gene questions and the like. But you got it. That was just fantastic. So, one more time, a round of applause. Dr.
Thank you so
Blackmore.
much.